Hello, everyone, and welcome to the start of the fall DFHCC Connect Science Seminar Series. I'm delighted to host today's speaker, Dr. Alan D'Andrea of the Dana-Farber Cancer Institute. But before introducing Alan, I have a couple of brief announcements. Firstly, for those of you who are new to this Zoom seminar series, please take note of the website that's shown on the introductory slide, shown here at dfhcc.harvard.edu/connectscience. There you can find both a list of future presentations and also recordings of all the previous seminars in this series. So it's a treasure trove of fantastic research. Secondly, any questions you have after Alan's seminar, please record them in writing in the Q&A window associated with this Zoom, not in the chat window. We won't see that, but the Q&A window is the way you get your questions up. And we will get through as many of those questions as possible at the end of today's presentation. Any questions that aren't answered will be sent on to Alan, who will then contact you to address them. So Alan D'Andrea is the Fuller American Cancer Society Professor of Radiation Oncology at Harvard Medical School, Director of the Center for DNA Damage and Repair, and also Director of the Susan F. Smith Center for Women's Cancers at the Dana-Farber Cancer Institute. Alan has a long and storied history in DNA repair and genomic instability. After receiving his medical training at Harvard Medical School, Philadelphia, and then completing a fellowship in pediatric hematology oncology at DFCI, Alan joined Harvey Lodish's lab at the Whitehead Institute at MIT, where he gained fame for cloning the erythropoietin receptor. He then joined the Dana-Farber faculty in 1990 and rapidly established an interest in the Fanconi anemia pathway. At the time that Alan began his research into Fanconi anemia, it was a very poorly understood set of genetic disorders. And it was really Alan's investment, sustained, intense investment over years, that converted that poorly understood clinical syndrome to its current status, where we understand the Fanconi anemia pathway to be a central component of the replication stress response, relevant to cancer initiation and cancer therapy. Alan's research has been recognized with numerous awards and prizes, including a 2019 Fanconi Anemia Research Fund Lifetime Achievement Award. Alan's interest in cancer therapy extends to all aspects of double-strand break repair. And today, we're going to hear about his work on an important gene involved in alternative end joining. So I'm delighted to welcome Alan's talk, Polymerase Theta Inhibitor Overcomes PARP Inhibitor Resistance. So Alan, thanks for joining.
Ralph, thanks so much, and I hope you can hear me. You can, okay. Right, so I'm very fortunate to have had such a articulate host introducing me. What a pleasant surprise. I heard about this talk last night, that there was an open slot today, so I was clicked to join it because I wanted to have this opportunity to present a relatively new, unpublished story. So here we go. So I'm going to tell you about polymerase theta inhibitor and how it overcomes PARP inhibitor resistance. So these are my disclosures. I will not be discussing any off-label use or investigational use in my presentation. Okay, so many solid tumors are homologous recombination deficient. And we're talking about ovarian cancer, breast cancer, pancreatic cancer, and prostate cancer. Those tumors sometimes result from germline or somatic mutations in HR genes. PARP inhibitors are known to kill HR deficient tumors. They do so by a mechanism of synthetic lethality. Homologous recombination deficient tumor cells become resistant to PARP inhibitors, often by restoring homologous recombination, perhaps by restoring cells' replication fork stability as well. PARP inhibitor resistance is a major problem in the clinic, and patients often respond very well to a PARP inhibitor, which is an oral drug, a relatively well-tolerated drug, but unfortunately after 8 or 10 months on the drug, patients often develop resistance and the tumor comes back. And there's really a major need for a drug which can overcome this resistance. So just one such tumor is high-grade serous ovarian cancer. This is a terrible disease. 27,000 women a year have ovarian cancer, high-grade serous. It's the 5th leading cause of cancer death in women. It's usually not detected until late stage. 75% of the cases are either stage 3 or stage 4. And this tumor typically responds to cisplatin, but then becomes cisplatin resistance. But it was actually the response to cisplatin which kind of led a lot of investigators to believe that this tumor often had defects in homologous recombination repair, the kind of repair mechanism that deals with platinum crosslinks. So it turns out that's true, that 50% of high-grade serous ovarian cancer has a defect in HR. As you can see in the pie figure on the right, there are germline and somatic mutations in BRCA genes and in other Fanconi anemia genes, which are underlying this cancer. Actually, interestingly, there's also nucleotide excision repair mutations and mismatch repair mutations as well. So very much ovarian cancer is a disease of DNA repair deficiency. There are multiple PARP inhibitors that have been approved. BRCA1 and BRCA2 mutations are not necessary nor sufficient for a response to a PARP inhibitor. There are plenty of cases of tumors, which you'll hear about now, where they're BRCA1, BRCA2 mutated, but the tumors are still very resistant to a PARP inhibitor. There's a tremendous need for functionally distinguishing true pathogenic mutations of BRCA1 and BRCA2 from benign variants. And there's really a need to extend the use of PARP inhibitors to this other 50% of squamous cell cancer, of high-grade serous cancers. So how do PARP inhibitors work? I know we have a diffuse audience, not a diffuse audience, I guess a disparate audience. What I'd like to say is the way this inhibitor is believed to work is through synthetic lethality. That if a single strand break occurs in DNA in a cancer cell, this single strand break can be repaired by PARP. PARP enzyme contains a zinc finger motif. It can bind to this single strand break and repair the break via base excision repair. But if one inhibits PARP with a PARP inhibitor, now this single strand nick is converted to a double strand break. And that double strand break often requires homologous recombination. So if the tumor is deficient in homologous recombination, the PARP inhibitor will kill that tumor. There's another mechanism of PARP inhibitor resistance, PARP inhibitor sensitivity shown here, that when the PARP enzyme binds to a single strand break, when the PARP inhibitor binds to the PARP enzyme, it can now trap the PARP enzyme in the chromatin. And this trapped lesion of PARP inhibitor bound to PARP often requires homologous recombination for repair. Hence, these HR deficient tumors are sensitive to the PARP inhibitor. Now, really what we want to talk about today is the mechanisms of PARP inhibitor resistance. And there are many such mechanisms. I'm only going to describe 5 of them here. So the most commonly touted mechanism of PARP inhibitor resistance is the following, that a tumor might have a mutation in BRCA1 or BRCA2, or perhaps another Fanconi anemia gene like RAD51C. And this mutant protein is dysfunctional, but in the presence of a PARP inhibitor, there could be a somatic reversion, creating a new open reading frame and now a functional protein. The cells become resistant. A second mechanism is the following, that in some cases in a tumor, BRCA1 promoter might be hypermethylated. These are obviously low expressing BRCA1 tumor cells, but in the setting of a PARP inhibitor, these cells can become resistant to the PARP inhibitor by upregulating or essentially demethylating this BRCA1 promoter. Another mechanism is the following, that a tumor cell might have a hypomorphic mutation in BRCA1 and BRCA2. And that hypomorphic mutant might encode a mutant protein with partial function, but in the setting of a PARP inhibitor, this mutant protein might be upregulated in expression, perhaps through an HSP90 chaperone mechanism. And this would result in an upregulation of the mutant protein and an acquired PARP inhibitor resistance. Now, all of these mechanisms result from a resurgence or a renewal of homologous recombination repair in the cell. And that's how the cells become PARP resistant. Recently, it's become clear that there's another mechanism of PARP inhibitor resistance. So homologous recombination deficient tumor cells can acquire resistance by knocking out this complex. So this is a complex of proteins called the Shieldin complex, which includes 53BP1 RIF1 Rev7, and these shielding proteins. This complex of proteins blocks DNA repair and resection, and actually thereby decreases homologous recombination. But a tumor cell exposed to the PARP inhibitor may knock out one of these proteins and essentially knock out this shielding complex, resulting in increased resection and renewal of homologous recombination. So a knockout of any of the proteins in this complex can cause PARP inhibitor resistance. In another mechanism demonstrated largely by Connor Claremont and Prabha Sarangi in my laboratory, they recently showed that in fact, some tumor cells can amplify a gene called the TRP13 gene, which encodes an ATPase TRP13. And this enzyme can release this shielding complex by essentially causing a conformational change in the Rev7 protein. And this release results in, again, an increase in homologous recombination and PARP inhibitor resistance. So tumors are very smart. What they're doing here is they're amplifying a gene whose protein product releases the shielding complex and activates homologous recombination. This was recently published in Nature Cell Biology. So the fifth mechanism, and this is really the mechanism we're going to deal with mostly today in my talk, is the following: that homologous recombination deficient tumors can become resistant to a PARP inhibitor by upregulating the activity of a different DNA repair pathway called the alternative end joining pathway. So you can see here in this schematic that if a tumor cell has a double-strand break, this double-strand break might occur as part of normal replication during S phase. This double-strand break will be resected, leaving these 3' overhangs. Now, the way this resected double-strand break is normally repaired is by homologous recombination, which is an error-free repair mechanism requiring BRCA1 and BRCA2. But if there's a defect in the tumor cell, no expression of BRCA1 or BRCA2, what happens is the tumor now elects to use a different DNA repair pathway called alternative end joining. In this repair pathway, which also occurs during S phase and also requires some resection, involves PARP enzyme itself, but also requires this very interesting polymerase called polymerase Q, which I'll also refer to as polymerase theta. So when alternative end joining occurs, and it's a very efficient way of performing so-called microhomology mediated repair, this is an error-prone repair mechanism. So when this pathway is active, this polymerase Q generates point mutations, generates small deletions and small insertions. And this mutational signature, known as signature 3, is actually a genomic biomarker for the hyperactivity of alternative end joining and the loss of HR repair. So we really need to have a drug now to overcome PARP inhibitor resistance. And there are a lot of labs and biotech and pharmaceutical companies working on this. So Raphael Sicoldi in my lab a few years ago now demonstrated a really strong synthetic lethality between homologous recombination and polymerase theta mediated alternative end joining. And he did a very simple experiment. He took a mouse, this was a Fanconi anemia D2 knockout mouse, which had a very mild homologous recombination deficiency and actually had a very mild phenotype. And he crossed this mouse with a mouse that had a mutation, biallelic mutation, polymerase theta. It had an alternative end joining deficiency. Again, a very mild phenotype. In fact, the only phenotype of these mice is they have increased micronuclei in their red blood cells. But when he crossed these mice, he found severe embryonic lethality, really proving that knocking out Fanconi BRCA pathway and knocking out Pol theta alternative end joining was a strong synthetic lethal event. This was borne out also through a CRISPR screen, which we did collaboratively with Steve Elledge's lab. And in this particular screen, BRCA2 mutant cells were exposed to a whole genome CRISPR library. And interestingly, guide RNAs directed against POLQ, or also against FEN1, which is another enzyme that's in the alternative end joining pathway. Guide RNAs against either of these gene products resulted in loss of alternative end joining and synthetic lethality. So this is really more evidence then that knocking out POLQ might be an efficient way of killing an HR deficient tumor. So how do we target Pol theta? Well, it's a big enzyme. It's a big gene. The gene encodes a protein of 2,500 amino acids. And actually, it has two enzymatic domains. The amino terminus of Pol theta has a Rad51 interacting motif and an ATPase helicase motif. Okay, I'll call it the ATPase domain. And then at the carboxy terminus of this large protein, Pol theta, is a polymerase domain. And we know now that this ATPase domain of Pol theta seems to be quite important in removing Rad51 from sites of abortive homologous recombination repair. Essentially, what this ATPase domain does is it cleans up a double-strand break to allow the polymerase to perform its MMEJ-mediated function. All right. So we went on to show this. Raphael went on to show that if he took cells now and he knocked out polymerase theta, he saw a net increase in Rad51 accumulation at the sites of double-strand breaks. In contrast, of course, if you knock out BRCA2, one sees a loss of Rad51 foci altogether. So this was really the first evidence in our hands that at least that knocking out polymerase theta resulted in this increased level of Rad51 accumulation. And then we think that this accumulation of Rad51 is actually toxic, and that's the mechanism by which the Pol theta inhibitor will kill an HR deficient tumor cell. To prove that further, what Raphael went on to do was he took BRCA2 deficient cells, which as you can see are very sensitive to PARP inhibitor, and he knocked out Rad51. And when he knocked out Rad51, these cells actually became resistant to a knockdown of the Pol theta gene itself. So this really said to us that Pol theta can kill, knocking down Pol theta can kill an HR deficient tumor cell by resulting in the upregulation of this toxic level of the Rad51 enzyme. All right, so this leads us then to all of the new unpublished work, which very recently, it's posted, it's not published, it's posted on bioRxives. And this is work largely of Jia Zhu, who's a postdoc in our lab, and a collaboration with Raphael Sacaldi, a former postdoc in my lab, who's now at the Institut Curie in Paris. So what Raphael originally started when he was with us was he decided to come up with a small molecule inhibitor of the ATPase. So here's the full-length Pol theta enzyme. It has the ATPase at the N terminus, the polymerase at the C terminus. He cut off the enzyme, so this is the first 1,000 or so amino acids of Pol theta. Here's the protein expressed on the Kamasi gel. And now, fortunately for us, this Pol theta actually has very strong ATPase activity in vitro. So he was able to do a small molecule screen up to 100,000 small molecules and found a very potent molecule in the first set of 25,000, which is an enzyme, which is a known drug, which is called novobiacin. Okay, novobiacin turns out to be a competitive inhibitor of the Pol theta ATPase. So what is novobiacin? This is what it looks like. Turns out novobiacin is a known antibiotic. It was used 30 years ago for the treatment of E. coli sepsis, so it's the antibacterial. The drug is actually quite well tolerated, and it turns out that the drug also has a relatively strong IC50 or low IC50 of 50 micromolar for the ATPase of Pol theta. It actually also inhibits, there are off targets, this drug also inhibits the ATPase of HSP90 and of topoisomerase 2, but at a much higher, more like a millimolar IC50. The reason I bring this up, this 50 micromolar concentration, is important because this is a readily achievable serum concentration in patients. And in fact, Paul Eder and others have actually tried to use novobiacin for the treatment of cancer patients, not knowing what the target was. Now we know it's Pol theta. And in fact, there were some extreme responders. There were some patients with metastatic breast cancer who responded to this novobiacin, but at the time those studies were done, it was not known whether or not those patients were BRCA deficient. And so because these studies were actually done before the identification of BRCA1 and BRCA2 genes. So what Gia went on to do, again, Gia, that's a very talented postdoc in the lab, was he coupled novabias in the drug itself to beads, which you can see the yellow hue of the novabias and conjugated beads. And he pulled down cell extracts, and he actually found that the novabias and the drug conjugated beads selectively pulled down the Pol theta enzyme, but actually did not pull down other known ATPases like the SMARCA ATPase or the CHD1 ATPase or the HSP90 ATPase. So there seemed to be a relatively strong binding activity for novabias directly for the ATPase of Pol theta. He then went on to do thermal shift assays. So here he took, in vitro, took the Pol theta protein itself and showed that the Pol theta protein itself binds to novabiasin, and when it binds to novabiasin, the protein is more stable and actually can be stabilized up to 46 degrees. And this kind of thermal shift stabilization is another way of showing that the drug is actually directly binding to the enzyme. Similarly, as a control, PARP enzyme, in this case olaparib, PARP inhibitor binds to the PARP enzyme and stabilizes the PARP1 protein. And then again, in collaboration with Raphael Sicaldi, what we did is the following experiment where we UV irradiated human cells in this case, and those cells now actually accumulate strongly the GFP tagged Pol theta at the site of irradiation. But if you add a PARP inhibitor, or if you add the ATPase inhibitor, novabiasin, you strongly blunt this recruitment of Pol theta to the site of DNA damage. And that's interesting because it means that the ATPase of Pol theta seems to be quite important for the actual recruitment step of the enzyme to the site of the DNA damage. So then in collaboration with John Tainer and Aleem Saeed, we did some modeling, and we were pleased to find that actually the novabiasin itself, which again is a known inhibitor of the DNA gyrase from E. coli, that this novabiasin drug actually models rather nicely in a groove in the Pol theta helicase itself. And in fact, if you turn it on its side, the model on its side, you actually see that the novabiasin is entering through one groove, and the ATP analog, AMP-PNP, actually is binding at another site. But based on this model, it's conceivable that the novabiasin, which is a long molecule, actually can inhibit the and block through a competitive inhibition, block the binding of the AMP analog. So of course, now that we knew it was a known drug and it had high affinity for Pol theta, we wanted to test this in cells and of course test it in vivo. So initially we tested it in cells. So Connor Claremont in the lab generated these BRCA1 deficient RPE cells. And these cells are very sensitive. They're null for the BRCA1 expression. There's no BRCA1 protein expressed at all. They're also P53 mutant. These cells are very sensitive to rucaparib, to a PARP inhibitor. But interestingly, we found that these cells, which are BRCA1 deficient, are also very sensitive to novabiasin. So of course, then we wanted to go right to in vivo studies because we were dealing with a known drug, a drug which can easily achieve concentrations of 50 micromolar in mouse models and in humans for that matter, which is the concentration that we needed to inhibit the enzyme. So the first thing we turned to was the well-known Yoss-Yonkers model. This is a model where it's a genetically engineered mouse model, which is K14 Cre BRCA1 TP53 a flox model. And these mice, when you knock out BRCA1 and P53, these mice develop mammary tumors, breast tumors, rather slowly over a period of 140 to 280 days. But once the initial mouse develops this tumor, this BRCA1 null tumor, this tumor can now be cut up into pieces and injected into the mammary pad of syngeneic mice. And we're able to generate now a colony of mice which contains this very aggressive BRCA1 deficient breast tumor. And over a period of 2 to 3 weeks, this tumor grows very quickly. So this offered an opportunity now to test whether or not novobiocin works in vivo. So to do that, in fact, here are the studies, it became clear that without treatment, this tumor would grow very quickly. Over a period of 10 days, and these mice would rapidly die from this large tumor. But if actually during this time instead, these mice are treated with novobiocin at a concentration that's given a 50 micromolar serum concentration, this tumor is clearly held at bay. And then when the novobiocin treatment is stopped at 35 days, of course, the tumor grows back. So this is really good evidence that the novobiocin is killing the BRCA1 deficient tumors, much the way a PARP inhibitor kills these BRCA1 deficient tumors. We performed some other xenograft studies with the drug at the same time. This was a xenograft model, an ovarian cancer xenograft model. This was a BRCA, a Fanconi F, so a mutation in one of the Fanconi BRCA genes, Fanconi F. This tumor is known to be HR deficient. The tumor actually grows rather quickly, but when this tumor in vivo was exposed to novobiocin, 100 milligrams per kilogram novobiocin twice a day. And by the way, this is an oral drug too, so the drug can be delivered either by gavage to the mice or by a peritoneal injection. The drug would prevented the rapid growth of this ovarian cancer tumor. So it works well in genetically engineered mouse models and in these xenograft models. So then the question became, if PARP inhibitor is failing now in the clinic, and there's a lot of patients who are developing PARP inhibitor resistance, would it be possible to add novobiocin to a PARP inhibitor clinical protocol? And in fact, we very recently, Jeff Shapiro is doing this work with us. Jeff and I recently received a clearance from the National Cancer Institute to conduct 2 clinical trials combining novobiocin with the PARP inhibitor. We wanted to show synergy here. And indeed, what we did was we took a BRCA1 deficient tumor cell. These are the null cells that Connor Claremont generated. These cells, if you had an increase in concentration of PARP inhibitor, you kill the cells, you get fewer colonies. But if at the same time you increase the PARP inhibitor concentration, you also add novobiocin, in this case at 100 micromolar concentration, one sees this synergy. The synergy is striking in that you can see here in this bar graph on the right, PARP inhibitor is an IC50 of about 2 nanomolar, which is a very potent drug. But in the presence of novobiocin, now the PARP inhibitor IC50 drops to 0.1 nanomolar. So it's a 20-fold increase in improvement in the IC50 by adding the 2 drugs together. And I think that this is important because PARP inhibitor, although it's oral and well tolerated, it does have some toxicity, as nausea, and it has, in bad cases, it actually can develop leukemia or neutropenia. This might be a useful way to actually limit the dose of PARP inhibitor by adding novobiocin concurrently. So can PARP theta inhibitor then kill a PARP inhibitor resistant tumor in vivo? That's probably the critical question. And indeed it does. First, we showed it does it in vitro. That is, we took tumor cells now, which are, this is a breast cancer, human breast cancer line, MB436. These cells are BRCA1 deficient. They're sensitive to the PARP inhibitor olaparib. But we've selected these cells in PARP inhibitor, and in this case, isolated a resistant line of PARP inhibitor resistant line, still BRCA1 mutated, but now PARP inhibitor resistant, because these cells have actually knocked out the Shieldin complex, and they knocked out 53BP1, one of the proteins in the Shieldin complex I spoke about earlier. That's a mechanism of acquired resistance. But we were very happy to see that these tumor cells now, which are olaparib or PARP inhibitor resistant, are still very sensitive to novobiocin. So novobiocin is as effective at killing these HR deficient cells as they are at killing the HR deficient cells with acquired PARP inhibitor resistance. So what about in vivo? So through work from Kalindi Parmer and Bose Kochaparakal and JB Lazaro in our Center for DNA Damage and Repair, we have we've been evaluating a number of PDX models. These are ovarian cancer patient-derived xenograft models. And these models have different characteristics. For instance, one of these ovarian cancer PDX models, the tumor cells, ovarian tumor cells, are still very sensitive to a PARP inhibitor. Turns out these tumor cells have a biallelic mutation in RAD51C, which is one of the Fanconi BRCA genes. And these cells have no RAD51 foci, which is the hallmark of an HR deficient tumor cell using Bose's RAD51 assay. And then here was another interesting PDX model. This was DF59. So this is a PDX model, ovarian cancer PDX model, where there's biallelic mutations in BRCA1. However, the tumor is very resistant to PARP inhibitor in vivo. And it turns out the mechanism of resistance is this tumor has lost 53BP1, lost both copies of 53BP1. So this tumor has lost the Shieldin complex effectively, and it has regained homologous recombination. And accordingly, this tumor is very resistant to a PARP inhibitor. You can see RAD51 foci are present here, consistent with the fact that this tumor has active homologous recombination. So we looked at these two models now, the DF83 mouse model and the DF59 mouse model, the HR deficient and the HR proficient model. And we do that, we were pleased to find that this tumor now, this ovarian tumor in vivo, was still, the HR deficient one was still sensitive to a PARP inhibitor. It was more sensitive to novabiacin, and it was extremely sensitive to the combination of novabiacin, the Pol theta inhibitor, with the PARP inhibitor. So much so that actually when the combination of novabiacin and PARP inhibitor were given, there was essentially no tumor evident in these treated mice. But the more interesting model is the one on the right then. These are the tumor cells now which have acquired PARP inhibitor resistance. They have a BRCA mutation in both alleles. They've lost the Shieldin complex. And these tumors actually are quite resistant to a PARP inhibitor. They're really resistant to the PARP because they've restored homologous recombination. And even though they've restored recombination, they still are very sensitive to novabiacin in vivo and very sensitive to the novabiacin with the PARP inhibitor. So this is really, I think, the first evidence that the novabiacin could be used in principle to treat a PARP inhibitor resistant tumor, and at concentrations of drug which are readily achievable in a human patient population. So what are the biomarkers for this? And this is rather interesting, that turns out when you take these tumor cells now from this PARP inhibitor resistant mouse and look at the accumulation of RAD51 foci. As I said to you earlier in my talk, when you knock out when you knock out polymerase theta, knock out the stripping activity of polymerase theta, now RAD51 builds up in the cell, and this RAD51 is toxic. If you treat the mice with a combination of novabiacin and PARP inhibitor, you have this even stronger increase in RAD51 accumulation. So these are all tumor samples taken right from the treated mice. So essentially then immunohistochemistry for RAD51 and the accumulation of RAD51 is a very convenient pharmacodynamic biomarker showing us that the novabiacin is hitting the tumor and causing this accumulation of toxic RAD51. In the HR deficient tumor cells now, these are the DF83 cells, which have a profound HR deficiency, you do not see this accumulation of RAD51. So we don't have a biomarker for these cells per se. Now, similar to this accumulation of RAD51, there's another pharmacodynamic marker, and that is the accumulation of phospho-RPA, the study recently done by Jia Zhu in the laboratory. And again, what we find here is the tumor, which has been treated with novabiacin, builds up the phosphorylation of RPA, phospho-RPA. And the combination of novabias and olaparib also gives this large accumulation of phospho-RPA. So the accumulation of phosphorylated RPA is tracking with the accumulation of RAD51. This is another useful biomarker showing us that the drug is actually hitting the tumor cells. Now, what is phospho-RPA? Phospho-RPA is a protein that builds up when there's an increase in double-strand break end resection. So increased RPA, as shown here at the bottom, correlates with increased double-strand break end resection. So now we seem to understand how this drug is actually working. That if you knock down 53BP1 or knock down the Shieldin complex, you get an increase in double-strand break end resection. Now those cells become very dependent on polymerase theta to block further end resection. So if you inhibit further end resection with the drug novabiasine, now you get even more double-strand break end resection, more accumulation of RAD51, and cell death. So we think that this phosphorylated RPA is a very convenient biomarker for demonstrating the increase in resection, which occurs in the, and particularly occurs in the presence of novabiasine, the Pol theta inhibitor. Now, what about predictive biomarkers? I showed you a pharmacodynamic biomarker. Well, it turns out Pol theta itself is expressed at very low levels. Pol theta it's one of the 15 human DNA polymerases. It's barely expressed at all on normal human cells. But in tumor cells which are sensitive to novabiasine, in tumor cells that have this increase in double-strand break end resection, these cells tend to build up the expression of Pol theta. Pol theta can be detected by qPCR assay, as shown here. So tumor cells which are sensitive to novabiasine have elevated Pol theta expression. So the target itself is upregulated, and that's a convenient biomarker for predicting which tumors are going to respond to the drug. And that can be done by qPCR. It could also be done by a Western blot. And now we're trying to work out a immunohistochemistry assay for detecting Pol theta expression. Okay, so to push this story further, Gia and our lab collaborated with Anina Farkila, visiting scientists from Finland. And what they did was they selected for a number of PARP inhibitor resistant clones to further validate our model. So what Anina and Gia did was they started with a P53 BRCA1 deficient cell. These are truly BRCA1 deficient cells. You see on the right, there's no BRCA1 protein expressed at all by Western blot. And they selected these BRCA1 deficient cells in the presence of PARP inhibitor. And they isolated a number of clones which are now BRCA1 deficient, but are resistant to the PARP inhibitor. And we've characterized a number of these clones. These clones tend to have loss of the Shieldin complex. They lose 53BP1, or they lose expression of Shieldin 1, 2, or 3, or they lose expression of Rev7. These tumor cells, which are PARP inhibitor resistant, are still sensitive to novabiasine, okay? And the R1 and R2 clones, here's the R3, R4 clones. These clones are resistant to a PARP inhibitor, but sensitive to novabiasine. And accordingly, all of these clones, which are sensitive to novabiasine, have some elevation of Pol theta expression, confirming that Pol theta elevation at a protein level on a Western, or this is the qPCR level, that elevated Pol theta expression, elevated expression of the target, seems to correlate with elevated sensitivity to the drug itself, novabiasine. Okay, and that's true. Here it is on the Western blot as well, that an elevation of the Pol theta protein seems to correlate with sensitivity to the novabiasine. And as I said, I think this is a convenient biomarker because normal cells just do not express Pol theta. We see it expressed in some cancer cells, and we believe the upregulation of Pol theta expression in cancer cells is going to be representative of novabiasine sensitivity. Now, what about tumor cells which have a perfect somatic reversion? When I'll tell you what I mean by a perfect somatic reversion. These are CAPAN cells. These are a pancreatic cancer cell line, which we use a lot in our Lustgarten program with James Cleary and Perry at Dana-Farber. These cells are very sensitive. They're pancreatic, BRCA2 deficient pancreatic cancer cells, very sensitive to olaparib, the PARP inhibitor. But these cells, when they undergo a perfect somatic reversion, where the BRCA2 gene has now been perfectly corrected, actually engineered to be a perfect correction, now these cells are resistant to the PARP inhibitor. And similarly, these cells, which have a perfect somatic reversion of the BRCA2 gene, are now resistant to novobiocin as well. So what this, and you can see on the far right, when a tumor cell like the CAPAN cells, which are BRCA2 deficient, they have high level of expression of Pol theta, consistent with this being a predictive biomarker. But when these cells are perfectly corrected by a perfect somatic reversion of BRCA2, they no longer express Pol theta on a Western blot, and now they're resistant to novobiocin. So this basically says to us that novobiocin as a Pol theta inhibitor will be particularly useful in the treatment of PARP inhibitor resistant cancers, ovarian, breast, pancreatic, and prostate cancers, which have a partial correction of homologous recombination. But if a tumor undergoes a perfect correction of homologous recombination and actually has a perfect somatic reversion of the BRCA gene, these tumors, unfortunately, will remain sensitive, resistant to either a PARP inhibitor or to the Pol theta inhibitor. But as we know from circulating DNA studies in patients who have BRCA mutations, somatic reversions are usually imperfect. Somatic reversions of a BRCA mutation do not result in a perfect conversion to a wild-type BRCA2 allele. They often result in truncations, splicing defects, which generate new open reading frames. And we believe that those mutations will still have sensitivity to the Pol theta inhibitor, and we're testing that now. All right, so back to this mechanism then of synthetic lethality and this increased RAD51 toxicity. What you can see here is taking some of these clones now, which are Pol theta sensitive, Pol theta inhibitor sensitive. This particular clone R1, it turns out it is sensitive to novobiocin. It's got a BRCA1 mutation. It's got acquired resistance to novobiocin. It's got acquired resistance to PARP inhibitor, but it's still sensitive to novobiocin. This tumor is sensitive to novobiocin. But when you knock out RAD51, now the cells become resistant to novobiocin. And this is, I think, more evidence that the mechanism of synthetic lethality is this accumulation of end resection and RAD51, which seems to be a marker for novobiocin sensitivity. And if the cell, so this might be a mechanism by which tumor cells become resistant to novobiocin is by down-regulating the expression of RAD51 protein. All right, so in the last few slides, all right, a couple of summations here. So what I've shown about mechanisms of synthetic lethality is, how does novobiocin kill an HR deficient tumor cell? Well, as we said at the beginning, if a tumor is HR deficient, now it really needs alternative end joining or microhomology mediated end joining, and novobiocin will block that pathway. But how does novobiocin kill an HR deficient tumor cell which has acquired PARP inhibitor resistance? Like, for instance, the acquired resistance might be from resulting from a knockdown of the shieldin protein or an amplification of the TRP13 gene. And the answer there seems to be that the novobiocin kills these cells by inhibiting the release of toxic RAD51. And so this accumulation of RAD51 becomes the mechanism of how the drug is working, but also a convenient pharmacodynamic biomarker that can be followed during a clinical trial. The conclusions of this then are that HR deficient tumor cells are PARP inhibitor sensitive, but can acquire PARP inhibitor resistance. And I gave you 5 examples of that at the beginning of my talk. That HR deficient tumors are sensitive to either a PARP inhibitor or to a Pol theta inhibitor. The tumors with acquired PARP inhibitor resistance can remain sensitive to a Pol theta inhibitor. That a PARP inhibitor resistant tumor can express high levels of Pol theta messenger RNA and protein, and this is a predictive biomarker. And that Pol theta inhibitor kills PARP inhibitor resistant cells by causing this accumulation of toxic Rad51 and this increased double strand break end resection that I described. So in the last few slides, I want to tell you about a new model system now that we're using for testing these Pol theta inhibitors. And the model system is high-grade serous ovarian cultures. So Sarah Hill, when she's a very talented fellow in my lab, now has her independent lab at Dana-Farber, Sarah figured out a way to biopsy tumors, ovarian cancer tumors, bring them to the laboratory, mince these fresh tumors up into single cell suspensions in matrigel, and then grow these organoid cultures. So these tumor organoids grow out in a period of about 7 to 10 days since after they were biopsied directly from the patient. So with these organoid cultures shown here is very early organoids, 7 to 10 days taken right from the patient. One can actually take these cells grown in matrigel and grown in a set of cytokines. One could do functional assays on these organoids. How sensitive are these organoids to certain drugs and drug combinations? Do the organoid cells have Rad51 foci or not? In other words, we can assess the HR activity in these organoid cells. Are the replication forks stable or unstable? We didn't talk about in my seminar today, but one can quickly assess replication fork stability in these ovarian cancer tumor organoids. And of course, one can do mutational testing on these organoids. So these organoids become a very useful tool for doing co-clinical trials, isolating a tumor directly from a patient, testing the sensitivity of that tumor cells in vitro, and at the same time giving those drugs concurrently to the patient herself, and seeing if one sees a correlation between how the organoid cells respond to the drugs and how the patient responds in the clinic. These organoids really do look a lot like the tumor from which they're derived. This is an organoid now, and histopathologically, these tumor cells look really just like the tumor cells from which they come. The organoid cells and even have lymphocytes present, which is hopeful that we can actually test the immunotherapy activity in the organoids while patients are being treated with immunotherapy agents in the clinic. One can do functional tests, as I said. One can take these organoids, this from a particular patient who had a BRCA mutation, or this actually patient that was BRCA1, BRCA2 wild type, but had a mutation in another Fanconi anemia gene. And there were no Rad51 foci and in the organoid, and the organoids were sensitive to olaparib, and indeed this patient responded to olaparib in the clinic. And this was another patient now who had a BRCA1 mutation in the germline, but this patient was not responding to a PARP inhibitor in the clinic, and accordingly the organoids derived from this patient also were not responding to the PARP inhibitor in the clinic. And indeed the organoids from this patient had Rad51 foci, showing that this tumor had actually resulted from a resurgence of homologous recombination, in fact, in this case, from the loss of the shielding complex. So now one could take these organoids and do tests in vitro. And this is very recent data generated by Arindam Bose, who's running our organoid facility at Dana-Farber. And what he showed, that the first organoid I showed you, which is PARP inhibitor sensitive, very PARP inhibitor sensitive, is also novobiasin sensitive. But the combination of the PARP inhibitor with the Pol theta inhibitor makes the cells even more sensitive. And so this is an example of how we can use a combination of drugs in the organoids and set this up as a co-clinical trial with the patient receiving the same combination. And this is, in fact, the more interesting patient now. This was the patient whose organoids were quite resistant to olaparib. You can see the organoids are very resistant to olaparib, the PARP inhibitor. They're also have a minor sensitivity to novobiocin, but the combination of novobiocin and the Pol theta inhibitor with PARP inhibitor really significantly causes a cytotoxic response in these cells. So we're kind of hopeful now that these rapid ovarian cancer organoids are going to be useful as a model system for looking at drugs and drug combinations and for testing these Pol theta inhibitors. And we're proposing using these organoids taken during the clinical trials that we're doing now with novobiocin and olaparib. Final summary then is HR deficient tumors are PARP inhibitor sensitive but acquire resistance. That HR deficient tumors are sensitive to either a PARP inhibitor or a Pol theta inhibitor. The tumors with acquired PARP inhibitor resistance remain sensitive to the Pol theta inhibitor. And the Pol theta inhibitor kills these tumor cells by accumulating Rad51. This work is done very close collaboration with Jeff Shapiro. Jeff and I co-lead the Center for DNA Damage and Repair at Dana-Farber. What we try to do through this center is identify new biomarkers for new drugs that are in clinical trials, particularly DNA repair drugs, and to employ these new biomarkers in clinical trials and using biopsies taken, pre and post-treatment biopsies from patients. And we're doing this now mostly for ovarian cancer patients, but also for pancreatic cancer patients through our Luscoden grant. Acknowledging people, many of the people in the lab, I briefly referred to the TRP13 data done largely, almost entirely by Prabha Sarangi and Connor Claremont. Lisa Moreau helped a lot with this work. And all of our collaborators in the CDDR, Center for DNA Damage and Repair, Jeff Shapiro co-runs it. Constantia Panteladou did all the work with the genetically engineered mouse model. These are some of the staff scientists I referred to their work. And a lot of this work is also done concurrently with Dipanjan Chowdhury's laboratory. And these are some of the former members of my lab. So I think I will stop there. Novobiocin work was done almost entirely by Jia Zhu, and I referred to most of his unpublished work here. So I'll stop there, and I see Ralph just popped up. So
great, thank you so much, Alan. That was really exciting and very, very exciting new development of your work on Pol theta. So in the spirit of the seminar series, I'm going to read out questions. I'm sorry that we can't sort of interact personally, directly. So the first question relates to the clinical reach of PARP inhibitors. Do you see a role for PARP inhibitors beyond HR defective cells as being potentially augmented by a drug like novobiocin, by a Pol theta inhibitor?
Yeah. Right. No, it's a good question. So PARP inhibitors, of course, are first ovarian, now breast, now prostate, pancreatic. Those are tumors that have bona fide HR deficiency. So 15 to 20% of pancreatic cancer actually has a mutation in the Fanconi BRCA pathway, which is astounding. But I think it's reasonable to think that one could extend the use of PARP inhibitors to colon cancers, for instance, by adding a drug which inhibits homologous recombination. So there's a lot of trials now, like for inhibitors of PI3 kinase inhibitors, for instance, or HSP90 inhibitors, or small molecules that actually can inhibit HR. And by inhibiting HR, they might synergize with a PARP inhibitor. And again, because PARP inhibitors are oral, well-tolerated drugs, they generally can be added pretty well to clinical trials. Most of the trials, combination trials are being done now, are combinations of PARP inhibitor with immunotherapy, though, because there's a real rationale for that immunotherapy, that PARP inhibitors can actually activate the STING pathway and confer a sensitivity to immunotherapy. So I think there's some hope that we can extend the use of these drugs to other tumors.
And a related question was coming from, again, focused on novobiocin. What do you anticipate the relationship might be between the ATPase inhibitor that you're working with and polymerase inhibitors of Pol theta, would you see those as being likely additive or fundamentally performing in the same way?
There was a lot of controversy as to which enzyme, which domain was a better target. But so far, best we can tell, inhibiting the ATPase domain or inhibiting the polymerase domain seems to give precisely the same answer. That is, and there's some unpublished work, so I can't talk about it, from a biotech company that suggests that polymerase inhibitors might do precisely what the ATPase inhibitors do. By blocking the activity of Pol theta, now one can treat a tumor which has acquired PARP inhibitor resistance. So I think there's no in vitro evidence that at all that says that targeting the ATPase domain is going to be any different than targeting the polymerase domain.
Right.
You know.
So one thing I'm wondering about in that regard is Rad51 foci, that if the ATPase domain is functioning as a sort of Rad51, functioning to strip Rad51 from toxic single-stranded intermediates, might you see a difference in the Rad51 focus response between, say, an ATPase inhibitor versus a polymerase inhibitor?
Yeah, well, that's a good question. We haven't done a side-by-side comparison. I guess I would predict that the ATPase inhibitor is more likely to activate this pharmacodynamic biomarker, this upregulation of Rad51, which is so conveniently measured by immunohistochemistry. The polymerase inhibitor might not activate that. However, I think both inhibitors, the ATPase inhibitor or the polymerase inhibitor, is going to result in an increase in double-strand break end resection, which is going to result in an increase in phospho-RPA. So that I think that mechanism will be similar, and that biomarker could be used in either case.
So our next question comes from someone of a mechanistic orientation, and it is in regard to Rad51, the enhanced Rad51 distribution that you see in NVB treated cells.
Yes.
The question is, have you performed ChIP-seq analysis to examine the genome-wide distribution of Rad51 in NVB treated cells? The question being whether you can detect Rad51 accumulation at sort of ectopic genomic sites.
Yeah, no, it's an excellent question. We've not done it. We don't know where it is, and the only evidence that it's really toxic is that, I showed you in a couple of slides, that when you knock it down, the Rad51 down, the tumor cells seem to do better, and they acquire resistance to novobiocin. But where that is occurring, I don't know. There's some very similar studies that were done by Gaurav Gupta at the University of North Carolina, where when he knocked down 53BP1 or components of the Shieldin complex, he saw an increase in double-strand break end resection and an increased accumulation of Rad51 when the cells were treated with, when the cells had a knockdown of Pol theta. He was doing an siRNA knockdown of Pol theta. He didn't have a drug to knock it down. So I think that seems to be a consistent theme. The way I like to look at it is, if you knock down the Shieldin complex, knock down 53BP1, RIF1, or any of the proteins in the Shieldin complex, you get increased resection. And now those cells really need Pol theta and MMEJ, alternative end joining, to stop further resection. It's almost like an active process to stop further resection. And if you inhibit that process, now you get long lines of resection, and that's toxic to the cell. So that seems to be a consistent theme.
Our next question is regarding cell identity. So this is a question about the ovarian cancer organoids. And the question is, what kind of cell type representation do you see in the organoid cultures compared to the native tumor?
Yeah, that's a great question. The earlier the organoid culture, the better. Okay, so if you do immunophenotyping on a very early, 3 or 4 day old organoid, you see a very diverse, distribution of T cells, dendritic cells, B cells, macrophage, and PAX8 positive tumor cells. But over time, the monocytes are the first to go. The distribution of cells changes. So I think that the organoid cultures will be particularly, if you really want to use them for studying immunotherapy agents, you have to use very early organoids because the cell distribution will change. But the tumor cells remain. The 90+% of ovarian cancer cells are p53 mutant. They have a selective. They grow very well in the organoids. In fact, you can add nutlin to kill the normal cells, any normal cells that are hanging around, and actually you can enrich for the tumor cells. So that's another trick that Hans Cleaver taught us.
So if we may finish, I'm going to ask one of my own questions, which is, I'm thinking about the RAD51 accumulation on, and as a toxic response. And I'm wondering whether one could perhaps probe that by looking at some of the known mutations in RAD51, such as, for example, the ATPase defective mutant that can't complete homologous recombination, or Agata Smogozewska's point mutant that shows a defect in the strand exchange property and underlying one of the Fanconi genotypes. I'm just wondering whether that might be a way to tease out this abnormal accumulation of RAD51 in the NVB-treated cells.
Yeah, well, we say the dirty little secret is it's hard to express Pol theta because it's such a big cDNA and such a big protein. We would love to do a side-by-side comparison, re-expressing wild-type Pol theta or helicase dead Pol theta or polymerase dead Pol theta, and seeing if we can rescue or not rescue the RAD51 foci. I think those are really good experiments. We have had trouble expressing the full-length protein. Now we're just expressing the domains one at a time, but we need to do more along those lines. The other obvious thing to do now, and I didn't even talk about it, is we're doing a lot of medicinal chemistry with collaborators to come up with more potent nova-biasin derivatives. Right now, and we're trying to co-crystallize, of course, nova-biasin. The crystal structure of the helicase is known. We have tiny little crystals, where, right now to try to and try to use some modeling to come up with even more potent nova-biasin derivatives. This is an important point. We know that nova-biasin has off-target activity. We're not making a claim here that 50 micromolar is not at the greatest drug in the world. It's going to hit other ATPases. But the fact of the matter is, nova-biasin is well-tested drug in the clinic to treat bacterial infections. We know so much about it, and it's it's relatively non-toxic. I think to that extent we can advance it. But the first clinical trial that Jeff Shapiro is going to do is with nova-biasin plus PARP inhibitor in patients who have PARP inhibitor resistant cancer. So we have the green light to do that.
Great. Well, Alan, thanks so much for this wonderful presentation. I think we all learned a lot. And thank you all for listening. And we look forward to other great talks coming up soon. So thanks, everybody.
Thank you.